So it says that it's preparing to live stream. You guys can let me know. Setting up. It looks like it's going well. Yep. I get a signal that we're okay, right? So it says that.
See you later, Esteban. I'm going to mute and stop the video so I can eat breakfast.
Lauren, I think we're okay, right? We're live? You're muted.
I think we are. It's tracking.
Okay.
Yeah.
Let's take 1 more minute. People are clicking in. And so after that, we'll do our usual introductions and 5 minutes to get us ready. My laptop crashed. I have no idea, so I changed laptops. Give me a second. This is a new one. So yes, I apologize for the inconvenience. My other laptop is restarting, but it looks like we're still live. Esteban?
Yes, it's still live over on YouTube.
Okay, well, that's good. That's good because I thought that the whole Zoom session had died. I'm glad it was just my local access, I guess. So I'll get started. I don't have my notes to proceed through because for those of you who are joining us now, the computer I was working from crashed. So I am now in a different computer where I don't have the files that I had opened for introductions, but I'll do my best. Good morning, everyone. It's a pleasure to be back. I missed the last couple of weeks of mine because it was spring recess here at Yale. So I took opportunity to go to the field in Argentina. It is a pleasure to be back. Let me quickly remind you of what we have ahead. So this is today, but next week we're going to have a talk on multiple paternity. Here we are. So next week we have Hannah Correa. You guys see my screen well? Lauren, we're okay?
Yes, we're good.
So next week we're going to have Hannah Correa and Steve Dobson present on why isn't there more multiple paternity in our model and phylogenetic analysis. And I had prepared a little bit of a slide describing how our talk from last week by Kaya Tombak speaks of how our community is growing. Kaya was part of a panel that we did, I think it was last year, on statistical and scientific inference. And that was just one talk that speaks to an interest of the fine organizers that every so often we tackle issues that relate to the methods that we use in the study of behavioral ecology and social evolution. So you may remember the panel we did with Kaya, Alba Garcia de la Chica, and Jeremiah. There was also a talk by Valentin Amrain from the University of Zurich discussing why we should move away from significance testing to estimation. And then there was one by Joe. Oh my goodness, Kaya, help me with Joe's last name.
Coleman.
Buck Coleman, that's another talk. So today's talk, which is going to be by Esteban Fernandez-Jurisic, who I introduce in a second, speaks to this theme that every so often we try to get into the FIND series. One which I think for me it's becoming more and more central. I think we're facing a time because of how communication has changed, where I sometimes feel like everything is reduced, everything is so simplified that we lose perspective of what defines what we do. And I will continue saying that what defines what we do is a set of methods, a way of making observations, a way of asking questions and answering them. And so many times that doesn't sell as well as communicating ideas of catchy, shiny results. But results are only, in my opinion, interesting and good if they're backed up. I mean, these being scientific results, they need to be backed up by what defines our job, which is doing scientific research. So today's talk very much, I think, speaks to that effect. Again, I lost my computer, so I don't remember exactly the title of Esteban's talk, but I know Esteban well. We've been talking about it for a while, so I know he's going to be discussing rigor in behavioral ecology and evolution. Esteban did his first degree in biology at the University of Cordoba in Buenos Aires, Argentina. It is, you may not know it, but it's the oldest university in the country, over 400 years old. Then he went on to do his PhD at la Universidad Complutense de Madrid in Spain, followed by postdocs in Oxford and somewhere else that are in Minnesota. He had a very short stay in California State. I guess he didn't like the weather in California, so he took a job in Purdue University, where he's been now since 2009. He's a tenured professor there. And what he's been focusing on, and the way he describes his research, which I want to mention because we may not be hearing a lot about, I don't know, about visual ecology and behavioral ecology, if the emphasis is going to be more on the rigor and methods in behavioral ecology. But he's been doing a lot of work on visual ecology, trying to understand and tackling questions at different levels of analysis with attention to the visual system of the animals that he's been focusing on, and those have been primarily birds. He is things that I noticed that I think make him different from us. He has a history of paying attention to methods and rigor. He's currently the code editor in behavioral ecology and sociobiology. He was until recently the president of a society. The acronym is SORTI, Society for Open, and he'll help me fill up the full name. It has to do with open research and open approaches to how we do and we communicate our science. He has close to 200 publications in all kinds of journals you can imagine, and he's also been consistently funded all these years in all the research that he's been doing. So I should have said he's from Argentina as well. But we only met like a few years ago. That's remarkable, right? I can't believe that it only happened a few years ago. It was a pleasure meeting him. And now there are many things that we're trying to do together. So it's my pleasure to welcome Esteban. Esteban, thank you so much for joining the FIND community. And please take it from here.
Thank you. Thank you very much. So I'll try to share screens. You let me know if you can see.
You're okay. But there you go. Yes.
So we're good?
I'm going to quickly move my laptop. You keep going. I'm going to silence and read.
Excellent. So, well, thank you so much, Eduardo, for such a nice introduction. And one of the things that I'm not going to talk about today is our main research focus in our lab, which has to do with addressing this question: how does visual perception affect behavior? I'll just tell you briefly that we try to be very interdisciplinary to address this question. We try to characterize the visual system, particularly of birds, but we have worked with other vertebrates using different anatomical and physiological techniques. We use a lot of modeling techniques to try to understand how birds are perceiving things in their brain, which allows us to make predictions that then we go and test using a bunch of different behavioral techniques and whatnot. So today I will not talk about this. Today I will talk about one of my hobbies. And what I've been thinking about a lot in the last few years is how can we be more rigorous in the research that we do in ecology and evolution. And a lot of the content of today's talk is not necessarily new, but it reflects some of the new trends that other fields, the biomedical sciences, psychology, and whatnot, have been dealing with, which is basically related to the crisis that the STEM disciplines are facing in terms of their ability to reproduce results. These started trying to quantify whether we can reproduce results in the psychological sciences a few years ago, more recently in the biomedical sciences, particularly with cancer biology. And the bottom line is that many of the papers that are published, they get a lot of interest, traction, and whatnot. When we try to reproduce them, we cannot. So the results are not the same. So this is concerning from multiple points of view. Obviously, the research rigor is probably the most important one, but there are also financial costs associated with this lack of reproducibility. Some papers have tried to quantify these irreproducible biology research, $28 billion a year. Other studies have said, well, that means that we're wasting a lot of resources in the research that we do. So in the biomedical sciences, they have tried to quantify this research waste. And more recently in ecology, people have tried to do the same. And even though the ranges vary quite a bit, it's about about 85% of the research we do ends up being wasteful research, right? For multiple different reasons. So the national academies in the US has been concerned about this. NIH has been concerned about this. NSF is very concerned about this. So there is sort of a global concern about what can we do to improve the reproducibility of our research. Even the media is now looking into science and saying, what's going on with you scientists? You need to figure this out one way or the other. So when we try to think about the causes associated with this lack of reproducibility, our brains, not everyone, but tend to go to the idea of fraud, right? And there is no question that there is scientific fraud. It's difficult to measure for obvious reasons. But believe it or not, it seems that scientific fraud is not the main culprit in this lack of reproducibility. And multiple papers have proposed that actually the most proximate reasons about these reproducibility crises are associated with common scientific practices. These are things that are part of our culture as scientists. These are things that we have been told by previous generations. I've been told by previous generations, right? And at some point, until I learned about all these problems, I didn't know, right? So these common scientific practices, this is just a summary. There are many, many, many more, are associated with the misuse of p-values when we run statistical analysis, the lack of reporting effect sizes, having underpowered studies, the lack of replication within studies, issues in the copying and pasting of data, the lack of reproducibility when it comes to statistical analysis, and a bunch of errors in terms of experimental design, estimation of sample sizes, and particularly with the interpretation of statistical results. I'm sure you have heard about the terms harking and p-hacking, right? Harking is something that we're dealing with a lot these days, which is coming up with hypotheses after the statistical results are known and selling the paper as if those hypotheses were developed before collecting the data, right? P-hacking is related to running a bunch of statistical analyses and then only reporting in our paper the ones that were significant and failing to report the ones that were not significant, and so forth and so forth. So one of the questions that I usually get when I talk about these is, well, sure, the biomedical sciences, but in ecology evolution, we're good, right? We're good. Actually, we're not. This is a wonderful paper from a few years ago that tried to survey researchers and get an idea of how much these questionable research practices have been used, right? And the incidence of, let's say, harking about 51%, p-hacking 42%, cherry picking the statistical results 64% are really worrisome. And this is something that we as a community need to become aware first and try to find ways of dealing with this issue. So in today's talk, obviously, I don't have time to talk about a bunch of these different things. So I'm going to focus on these, the ones that are underlined in red. And what I'm going to do is I'm going to take, and I want to be very explicit about this, I'm going to take the frequentist perspective. Not because I have anything against Bayesian analysis. Don't worry about it. Please don't misinterpret me. But I'm going to go into the frequentist side of the business because many members of our community still use frequentist statistics, right? And one of the arguments I'm going to make is that maybe we're not using frequentist statistics the way we should be using frequentist statistics. So I'm going to focus on frequency statistics. By the way, as a side note, if you're interested in the sort of statistical wars between Bayesian and frequency statistics, I highly recommend this wonderful book by Deborah Mayo that talks about the back and forth between these so-called two camps. But that's an aside. So frequency statistics, within that, I'm going to be talking about hypothesis testing, right? Statistical inference and hypothesis testing. So I'm not talking about descriptive statistic. I'm not talking about estimation. I'm not talking about developing predictive models, right? So it's only about hypothesis testing. And you will see this figure so many times during this talk that you're going to get tired of it, but it has a purpose, right? So the idea of this figure is that these are the four, there are more, but these are four key components when we do frequency statistics when we're trying to test a hypothesis, when we're trying to do some kind of inference around them. So you know about the sample size, you know about the significance level. I'll talk about type 1 and type 2 errors in a few minutes. You know about the power of statistical analysis, and you know about effect sizes. But just to make sure we're all on the same page, effect size is a standardized way of measuring the difference between something that we measure. In this case, if we have a control and intervention and we have a dependent variable, right? The difference between these two is small, so we say it's a small effect size, but the difference could be large, and we say that's a large effect size, right? There are different types of effect sizes. We don't need to go into all these, but just to give you some clear definitions from the get-go. So what I'm going to do is I'm going to tell you now, instead of at the end, the take-homes of the talk. So take-home number one, these four elements are related, big time. Actually, they're mathematically related. That's the first take-home. Take-home number two, these four elements bound what we call the space of inference. Not just the p-value, all of them. So if we just get the p-value and we try to make any kind of inference from our statistical results, we're going to run into serious trouble. So we need these four elements to understand our space of inference. And the third take-home is our statistical results should be interpreted relative to the space of inference that these four values, these four components determine. So those are the three take-homes. Now let me try to work them out somehow logically, right? So in talking to you, to some of you today, and in informal conversations with other members of the community, there is no question that there is an obsession with the p-value, right? Is it significant? Is it not? Well, what are the consequences of being significant or the consequences of not being significant? If it's significant, we're all happy. If it is not significant, we're all very sad, blah, 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 right? So that obsession is present everywhere, right? So we, in some way, we convey to the new generation of scientists the idea that depending on the p-value, your research will go in this direction or that direction. And that has upset many members of the scientific community to the point that some have gone out and say, stop. We need to stop using statistical significance because it's really generating a negative effect in the way we do science. And that is a good argument. It is a really good argument. The problem is that there are other voices in the community that have expressed alternative viewpoints. And one of these viewpoints, which I'm going to be talking quite a bit today about, is, hey, there is no problem with the p-value. The problem is how we use and apply the p-value. And that's sort of the theme of this talk, that you know, we shouldn't get upset with the p-value. We should get upset with the way we are using that p-value. That's point number one. Point number two, maybe if we use the p-value correctly, along with the other three elements that I mentioned to you before, maybe we do have a framework to make some inferences. And that framework, I'm not arguing that it's better than Bayesian or worse. No, it's just one framework to run and, you know, to make some inferences from it. Okay. So going back to this, right, these four elements, when we're trying to make some kind of inference, are included to some degree in a version that you can see on the screen of the confirmatory method, in which we develop a question. From that question, we generate some kind of hypothesis and some kind of prediction that we then go and test with some kind of statistical analysis. Okay. Within the idea of frequency statistics, I want you to pay attention to this big arrow, which is related to the question. So, and the concept I want to transmit here is that a scientific question is a complex endeavor, or trying to answer a scientific question is a complex endeavor. And within frequentist statistics, running a single study is not enough to answer a scientific question. I leave that concept there. I'll get back to it in a few minutes. Okay. All right. So the other idea that is important to keep in mind is that when we're doing inferential statistics, we have a population, but we don't have enough resources to sample all the subjects in that population. So we have to take a sample of that and then make an inference relative from the sample relative to the population. Right? A sample, a study. Those are ideas that I want to communicate as background. All right. So the other important thing here is that we develop in the confirmatory method these hypotheses from which we derive predictions. Then the statistical hypotheses and the statistical predictions are not the same as the biological hypotheses and the biological predictions. We have to be a little bit careful with that. In fact, the statistical hypotheses and predictions are most associated with the biological predictions of our study, right? All right, so the framework that we tend to use within frequentist statistics is the null hypothesis significant testing framework. This is a framework that is it's a hybrid between the Fisherian and the Newman-Pearson frameworks, right? And the idea of that framework is to make some kind of dichotomous decision. So if you have a question like, is the treatment significantly different from the control, right? So we make a decision, yes or no. Is the relationship between X and Y significant, right? The answer it would be yes or no. And that answer, that dichotomous decision, is based on the p-value that comes out of the statistical test relative to a p-value threshold that we as scientists define. So as you know from basic statistics, we have the statistical null hypothesis, which is usually phrased as a lack of effect. We have the alternative hypothesis, which is phrased in terms of the outcome of interest, right? And that alternative hypothesis is what will probably match to a certain degree the biological prediction within the confirmatory method. Then we run a statistical test, but the test is against the null hypothesis. It's not against the alternative hypothesis. And that test estimates a probability value, the so-called p-value, that then we compare to the threshold to make that dichotomous decision I told you about before. All right. So here we have our area, right? So we have the threshold. Usually we tend to choose 0.05, but you can change that, so that's no problem. And then our statistical test gives us a p-value of 0.147, which is higher than that threshold. So in that particular case, we're going to fail to reject the null hypothesis. Failing to reject the null hypothesis has nothing to do with the alternative hypothesis. So failure to reject the null hypothesis is just about the null hypothesis. In the case of another statistical test that gives us a p-value of 0.021, we compare it with the threshold and it's lower, so then we reject the null hypothesis. Rejecting the null hypothesis, as you all know, doesn't mean that we support the alternative hypothesis, because under this framework, it's just testing, the test just runs relative to the null hypothesis. So then the question is, what is really the meaning of the p-value coming out of a statistical test? So I'm going to show you the book definition. So that p-value, this one right here, is the probability of observing the collected data, not other data, just the collected data, in one sampling event, assuming that the null hypothesis is true. That's it. And this has nothing to do with the probability that the results have occurred by chance, has nothing to do. So that's what a p-value is. So now I want to bring some of the things that I was telling you about before to sort of a bigger picture, right? So when we run a statistical test and we get a significant result, it's not a big deal, in the sense that we cannot generalize that result to multiple populations. Remember, we are inferring from the population that we're sampling, that's it. And if that population happens to be in Indiana, we cannot make inferences about populations in Tennessee. That's all that p-value is. So I think one of the misconceptions about the p-values is that it has been used to make generalizations that the p-value cannot promise, because this is all what the p-value is about, okay? In fact, at the core of frequency statistics is that we are supposed to replicate that study that we did in order to answer fully answer a scientific question. So a single study with a significant p-value doesn't mean much, because frequency statistics assumes that we're going to replicate that study multiple times to eventually answer that question, right? If you're interested in this, I encourage you to go to this website. It's a lovely simulation. It's a p-value intuition pump, in which you simulate the sample size. So this is the difference between a treatment and a control, let's say. You simulate your sample size, then you estimate your difference between groups, which is basically the effect size. And then you run that study as many times as you want. In this case, I chose to run it 100 times. So then you have the distribution of p-values after running that study 100 times. So what you can conclude, and this is the 0.05 threshold, what you can conclude is that of the 100 replications, only 11 led to significant results. The other 89 were non-significant. So then the question that we have to ask ourselves when we run statistics is, and we get a significant p-value, is is that p-value here? And if we don't replicate the study, we will never know. In other words, okay, we got significant results, but is that a fluke or is this part of a pattern? And one of the things that we're not probably thinking too much about is replicating the studies that we did. So a group of researchers asked that question: how much ecologies are replicating studies? And here's the answer, at least until 2019. So the frequency of replications, based on what they measure, right, was the percentage, sorry, not the frequency, the percentage of replication was 0.023. This is not the proportion, this is the percentage. So we, as ecologists, are not doing enough replications. So we need to keep this in mind when we, again, go and use the p-value to make generalizations about a biological phenomenon. We shouldn't be doing that, right? And that doesn't discredit the p-value, right? It's just a little bit of context for us to reach conclusions about our research. When we make a dichotomous decision like the one that I described to you before, we can be wrong, right? And that leads us to the idea of the type 1 and type 2 errors, right? So here you have the truth that the null hypothesis is true or the null hypothesis is false, and we don't know this, right? And this is what we know. This is the outcome of our statistical analysis, which is fail to reject the null hypothesis or reject the null hypothesis. We want to be correct, right? But we are not necessarily correct. So we can be committing a type 1 error, which basically is rejecting the null hypothesis when it actually is true. And actually that type 1 error, or alpha, is the threshold that we choose. So when we're about to run statistical analysis, we need to say, what is going to be our threshold? Is 5%, is 1%, whatever percentage, right? That's the alpha that we choose. But then there is the other type of error, which is the type 2 error, or beta, right? Which is when we fail to reject the null hypothesis when actually it's false. So this can be equated to the false positives for the type 1 error and the false negative for the type 2 error, right? But the important thing here to really become aware is that we are allowed as researchers to choose our type 1 and our type 2 error. We have to justify whatever we choose, right? But we're allowed to choose that, even though we have ingrained in our brains that it is all about that alpha 0.05. We can change that. We just need to justify it. So one of the things I told you about these 4 elements is the power of a statistical test, which is basically 1 minus the beta, which is the type 2 error, right? Again, is the probability of correctly rejecting the null hypothesis when it's false. So from a statistical point of view, we want to run statistical analysis with high statistical power, because then we'll have a good chance of detecting effects when they're present. So if you have a statistical test that has a power of, let's say, 15%, then more or less you have a 15% probability of detecting a significant effect. So then we need to think about these from the perspective of, okay, so should we run a study using inferential statistics? We can run the study using descriptive statistics or whatever. Should we run an inferential study with a 15% probability of detecting a significant effect? And I leave up to you to think about the potential answer to that question, right? But the other side of the coin of the statistical power is that, well, studies could have really good power, but studies can have low power, and consequently they're considered underpowered studies, which are studies that do not have sufficiently large sample size to answer the research question of interest. So the follow-up is, in general, studies are good or are underpowered? And unfortunately, the answer is that they're mostly underpowered, and that has been looked into throughout the biomedical sciences, which generates a lot of issues, but also in ecology and evolution, right? So people that looked at the studies that we do, they found out that we're running studies, inferential studies, with really low power. What are the consequences of running a study with low power? Well, there are many. The first one, based on the definition of power, is that we have a lower chances of detecting a significant effect. The second one, which is very interesting, is that if a significant effect is found, some people tend to think, oh, then we're good. I don't need to worry about power. No. You still need to worry about power because there is a higher risk that that significant effect might be a fluke. And that has been shown in multiple different studies. This is one of them. Another detrimental consequence is that when we try to estimate the effect size of that particular study, if we have low sample size, then there is more variation. So this is sample size. This is the effect size estimate. With lower sample size, there is more variation in our estimate of the effect size, which lowers the accuracy of our ability to tell how different two things are, in terms of that particular parameter, right? The other problem with small or underpowered studies is that they tend to overestimate the effect sizes because of publication bias. And publication bias is basically, if we get a study with non-significant results, we tend not to submit it. Or if we submit it, the chances of a journal accepting that study go way down, right? So what happens is that studies with low sample sizes that get published tend to have a higher effect size estimate because of that publication bias relative to studies with high sample sizes that have a more accurate estimation of that effect size. Is this a problem in ecology and evolution? Again, a recent study published last year found that yes, it's a huge problem. We are publishing studies in ecology and evolution that have these exaggerated effect sizes that in many cases is not the only reason, are connected with these underpowered studies. So going back to these 4 elements, right, that we talked ad nauseam now. So one of the things that is going on these days, particularly in areas of the psychological sciences and other disciplines, is that they're trying to encourage scientists to justify their sample sizes, particularly when it comes to inferential statistics. And many journals, including journals in ecology and evolution, are making sample size justification mandatory, right? So we have to declare the reason behind our sample size estimation, whether we like it or we don't, right? And there could be many reasons to do that, right? But the important thing here is that based on that sample size justification, then we can get an idea of how informative the data that paper is trying to share is, right? There are different types of justification, right? We don't have time to go over all of them, but the one that I want to pay some attention to at the end of this talk is the a priori power analysis of the idea of power analysis, right? So a rigorous way of justifying sample size is to run a power analysis, as you all know. There are different types of power analysis: a priori, compromise, a posteriori. As a side note, a posteriori power analysis have 0 value because basically they're a mathematical transformation of the p-value of our statistical test. So there is no point in running a posteriori power analysis. If an editor ever asks you about running a posteriori power analysis, you can find online resources to justify statistically why that should not be the case. But that's a side note. So I'm going to focus on a priori power analysis, which is the one that we usually tend to run, right? So the goal is to estimate our sample size. In order to run an a priori power analysis, we need to enter the type 1 error, the power of a statistical test, and we choose those 2 things. And we also need to measure an effect size. And that could be estimated from different sources of information. And the output will be the sample size. And that's usually the way we tended to report a power analysis. Oh, we ran a power analysis and we needed a sample size of 25, period. That's not appropriate, right? So the proper way of writing the results of a power analysis is, and this is just one example, we found that we need 138 samples to detect a significant effect size, and we need to establish what do we mean by significant, to detect a significant effect size of this much. So we need to have an estimate of the expected effect size with a 95% power. So in the reporting of the power calculation, we have these 4 elements that I told you about before. If some of these elements are not present, that a priori power analysis has very little inferential value, okay? And one of the reasons is because these 4 elements altogether provide the bounds of what we call the space of inference, okay? And it's very difficult to portray 4 dimensions in a 3-dimensional graph. So I just plotted here 3, right? Sample size, effect size, and power. But this space of inference is fundamental for the way in which we're going to interpret the results of any frequentist statistical analysis that we run. And the argument is that if we don't have a good estimate of that space of inference, any interpretation of our statistical analysis is going to be fraught with a lot of challenges, okay? So how can we think about this space of inference, given that, we're talking about 4 dimensions and our brain might be a little bit limited to think about those 4 dimensions at the same time? So what we can do is simply, we can think of 2 dimensions, right? We can vary these 2 dimensions and we can keep the others constant. So that's what I'm going to do here to explain to you a little bit about this space of inference, right? So let's say that we vary the estimated effect size that we're going to represent with these squares, right? So if the square is bigger, it means that the effect size is bigger, right? The difference between the treatment and control are larger and larger. And then we're also going to vary the estimated sample size, which we will represent with these circles. And this basically means that the bigger the circle, the bigger the sample size, okay? So we're going to keep the type 1 error and the power constant, and the ratio will be 1 between type 1 and the type 2 errors, right? And then one of the things that at least helps me to think about the space of inference is to try to think of this analogy with different types of microscopes, right? So imagine that one of the things that one of your research questions is, I want to detect in a sample the presence of this organism. Well, do I need an electron tomography microscope or a cryo-SEM to do that? No, you just need a stereo microscope. That's all you need, right? So you don't need to spend a lot of sampling resources to detect these. But what if your question is, I want to detect viruses that are inside of these organisms? Can I use a stereo microscope? No, you won't be able to detect them. You need to use a transmission electron microscope to do so. So that's what this idea of the space of inference allows us to do, because now we can transfer to these. So if we want to detect a very small difference between the treatment and the control, that will be a very small effect size, we need a lot of sampling size to do so. But if we're trying to detect a huge difference between the treatment and the control, with a small sample size, we might be okay. And that assumes these levels of confidence, right? A type 1 error of 5% and a power of 95%, right? If we change these 2, obviously this relationship might change a little bit, okay? So one of the problems that we have is that sometimes we want to detect a very tiny effect size, but we say, well, but we only have resources to have these many samples, so we should be okay. No, we're not okay. And the reason we're not okay is because we cannot, with this sample size, with this microscope, we cannot detect this small effect size, right? And we can only detect this large effect size. But by not being able to detect that small effect size, we might be having a sort of misalignment between our sample size, the effect size that we want to detect, and the experimental design. So we need to be thinking about these questions at the experimental design level. That's the point that I'm trying to make here. All right. So it is recommended that we establish our space of inference to understand what we can and we cannot conclude from the data we plan to gather. And this idea has to do with, hey, I got non-significant results. What does that mean? Well, it depends on where you are. So you got non-significant results, but you match these two. Well, what it might mean is that there might not be a biological effect with this confidence. But if you're mismatched, then you don't know. It might be that there is no biological effect, or it might be that you don't have the large enough microscope to detect that effect that you wanted to detect. And you can make the same argument when you find significant results. The exact same argument. You found significant results, but did you have the right microscope to find those significant results? No. Well, it might be a fluke. But if you had the proper microscope, well, then you have this confidence established by these two parameters that you found a significant effect. That's the idea here. Okay, so we need to do a better job at reporting our a priori power analyses. There are many publications that help us on exactly what we need to consider. Here is a list. We don't have time to go over that, but you get the idea that in order to do a proper a priori power analysis, we need to really define those four elements that I told you before. So hopefully I made the case, maybe I have not been able to convince you, but I made the case that just thinking about the p-value in isolation doesn't make any sense when we're trying to run inferential statistics under the statistical framework that I told you before, because these things are connected. And what people are trying to say these days is that, hey, this conversation needs to happen before, when we are running, when we are designing our experiments. And the way I was taught to design an experiment, and it's a bias that I have that I'm trying to unlearn it, is, hey, how many samples so we get a significant result? That's the way I was told to design an experiment. But what the community is trying to, or members of the community is trying to encourage us to do is we need to shift that viewpoint. So, and the idea is to design experiments not to detect a significant effect, but to design experiments to detect an effect size of interest with the researcher established levels of confidence. And those levels of confidence, again and again, are set by the type 1 and the type 2 error that you can change, right? So it's about the effect size of interest rather than whether it's significant or not. That's the shift in culture that we're trying to engage, right? So, and that's sometimes difficult to unlearn. And you know, I'm sure that some of you have been exposed to G-power, right? And when you run a power analysis with G-power, right, you're asked to put your type 1, the power, you know, a few other things. But then you get to the effect size window, and then you get these effect size conventions. Small, medium, large, right? And then you're like, oh, you know, I need to submit these to my ICAG in 10 minutes, whatever. Yeah, I think we'll get large. It's okay, right? And then you click large, and then you get a sample size, and then you're done. Well, the problem is poor Cohen, right? When he proposed these conventions, if you go back to the textbook, actually, he's discouraging people from using these conventions. And what he says is, if you have a situation in which there are no published papers, like nothing, I guess you could use these conventions, but please don't. What people have done is conventions. I like it, large, done. So that in some circles of the community these days is considered a questionable research practice. just so you know. Not everybody, but some people consider that rush into putting some kind of effect size a questionable research practice. And to some degree, it makes sense, right? Because the conventions vary tremendously depending on what your research question is. And that makes sense because biological phenomena are supposed to vary in their magnitude depending on what are you asking. You know, are you talking about mate choice? Are you talking about anti-predator behavior? Are you talking about social cohesion? Blah, 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 blah. The effect size is going to be different depending on what your research question is about. So using effect size conventions the way you know from Cohen's book is highly discouraged these days. And I think, and I try to have these conversations with as many people as I can. I think we need to have deeper conversations about effect sizes at the moment in which we're designing our experiment. And this is not easy because at least my generation has not been trained that way. My generation has been trained like, make sure it's significant. Okay, got it. I need to make sure it's significant. But I think what we need to do is sit down and start thinking, well, let's not think about significance first. Let's think about what kind of effect sizes I'm thinking about. And that's a conversation that it seems to me ecology and evolutionary biologists might want to have more often. I mean, I'm sure people have that, but I think we need to encourage that conversation to a larger degree, okay? So there are different ways that we can use to guide that conversation about the effect size of interest. I don't have time to go over all of them, but I'll just mention a few of them, right? So one way, and if there are grad students listening to this might be something you might be interested in doing. One way is that if you have a research question that has some history, it doesn't need to be long history, but that has some history. One of the things that you could do is you could go back to the literature and establish the different effect sizes for that particular question, right? And then get a distribution of the effect sizes in your research area. And then from that distribution of effect sizes, you estimate the 25, 50, and 75 percentiles. And then there you go. You have, for your research area, the small, intermediate, and large effect sizes in your area. And you can use those effect size values in your a priori power analysis because it's going to be more informed. These are the sort of effect sizes that we are expected to find in that particular, for that particular question. Another thing you could do, which actually it might be even better, is to go and look at whether your research question has had any kind of meta-analysis. Because then what you can do is you can use the overall effect size and the confidence intervals of that overall effect size that then you can input in your power calculations. One thing to keep in mind here is that when we use these estimates from a meta-analysis, we're supposed to run 3 a priori power analyses. One with the mean effect size, one with the lower 95% confidence interval, and one with the upper level of that confidence interval. That is going to give us a more accurate picture of this variation in our space of inference depending on the different types of effect sizes. And I think we could do is we can establish an expected effect size from theoretical models. And I think this is something that for behavior ecologies, we should be able to do this more frequently, but we don't have these sort of this practice in our field, right? So this is, I took this from a paper, right, that has different hypotheses relative to a particular phenomenon, and these different hypotheses lead to different estimates of that effect size, right? Well, then we can use those estimates in our a priori power analysis. Another thing we can do is we can, if we're limited by our sample size, we can estimate the minimal statistically detectable effect size. In other words, is the smallest effect size that, if observed, would be statistically significant given that sample size. we can run a sensitivity power analysis in which, again, if we're limited sample size wise, we can get an idea of how much sample size we can estimate, we can detect relative to the power of our statistical analysis, and so forth and so forth. And one of the things, and with this I finish, one of the things that it seems to be the sort of the future in these a priori power analysis is an idea that has been used in the biomedical sciences for a long time. And it's the idea of the smallest effect size of interest. So the key question is, which effect sizes would be too small to matter from a biological point of view? So we're measuring something, right? And we expect to find a difference. We hope it's significant. No. That's not the right frame of mind. The idea is it could be very small, at which point of small, we should say, I don't care. You know, it might be significant, but it's so small that biologically, it shouldn't make any difference, right? So the process of coming up with the smallest effect size of interest, actually, the first step is to determine the meaningful effect size of interest, which is the expected effect size that would optimize the efforts of applying any kind of intervention. And usually this is the from a cost-benefit analysis, right? So then from the meaningful effect size of interest, we can jump into the smallest effect size of interest, which usually is smaller than the meaningful effect size of interest. And this is, what is the bare minimum improvement after applying any kind of intervention that would justify some kind of effort, even though it's not the optimal effort, right? And this idea has been used in the biomedical sciences quite a bit, and hopefully this example gives you a sense of how easy it is to think about it in that context, right? So imagine that you have a treatment, right, and you have a control, and you find a significant effect between these two, which might be associated with a certain effect size. But if the patients don't feel improvement, even though you found a difference, it doesn't make any, you know, from the perception point of view of the patient, I'm the same. So that's what we're talking about with the smallest effect size of interest, right? And in medicine is the minimally clinically important difference represented by the smallest improvement considered worthwhile by a patient. So the biomedical sciences have these really well worked out. And I think what we need to do is to try to start thinking about these in the field of animal behavior and behavioral ecology. Final things, you know, we should also think deeply about G-power. G-power uses analytical formulas to run power analysis, which is totally fine. It does encourage using conventions in an indirect manner, and it's not very reproducible, although you can make it reproducible by taking screenshots and whatnot, right? I think we should also consider alternatives like the using simulation-based power analysis, and this works pretty nice because if you have a complex research question and things like that, then based on your estimated effect size of interest, you can simulate data sets, you can run in loops, right, test for statistical significance, and then you can calculate the power in a very easy way, right, by doing that. And this is something that more people are starting to use. I think we should make our power analysis also reproducible using R and using code. For some very complex statistical analysis like mixed models, there are some new packages, some of them not so new, that can help us navigate these because sometimes it's a little bit complex, right? And I just want to end with this idea from this paper that improving the power of our statistical tests through all the things that we discussed today can have a huge positive effect in increasing the reproducibility of our scientific studies by improving our ability to make inferences and also our ability to obtain accurate estimates of the effect sizes that we are so interested in. The paper these days on justifying sample sizes is this one, is open access, so I encourage all of you to use it. I'm a big fan of Dan Laken's or Lakens, I don't know how to pronounce it, work. And if you're interested, I encourage you to read his recent textbook. It's open access, right? And basically what it does is it gives us a lot of ideas on how to properly use these statistical analysis to improve our statistical inferences. And with that, I thank you very much. If you have questions about running your power analysis or mixed models, I love mixed models, just so you know. I'm happy to chat. So if you want somebody to bounce ideas, just email me and we can connect via Zoom. Okay, thank you very much.
Thank you, Esteban, for a very interesting presentation. So for those of you who are new to FINE or need a little bit of a reminder, the way we run now the next hour of questions and answers, which has become really a part of an integral part of the FINE seminar series. We all value this hour much. I ask that you type a question mark in the chatting window. And if you're a student, postdoc, I think we're using just the letter S. That will set you apart as from all of us, the senior faculty. We give priority to the young voices. We want you to participate and feel comfortable joining us in asking and answering. So please don't be shy in posing questions. So just again, type a question mark in the chatting window, and I'll be calling people in the order they posted their questions. And after that, I'll open it up to the whole community. So we already have a question. Oh, yes, please don't forget, because we enjoy getting to know each other. Even if you feel like you're a regular, some of our new participants may not know you. So please very briefly state the institution where you are and the country. Sometimes we assume that people will know where our institution is. Kaya Tombak, you have the first question.
Hi there, I'm a postdoc at Purdue, Kaya Tombak. I have a question about post hoc analyses. I understand why it doesn't really make sense to do a post hoc power test. But let's say you've done your study and you didn't know about this new analysis method that you want to apply, and you want to assess whether you have a good enough data set to do it. I read Gelman and Carlin study on post hoc analyses that aren't on power, but they're more estimating type S and type M error. So type S being the probability that the sign of the effect that you discovered is wrong, and type M is the probability that the magnitude of the effect is wrong. I kind of liked it at first glance. I'm not sure if you've seen it or if you've already dealt with this kind of post hoc analysis, but I think in principle, at least, it seems like a nice way to put some parentheses around your results that you found.
Absolutely. Great question. One of the upsides of the reproducibility crisis is that there are a lot of people out there, or there have been a lot of people out there developing new quantitative approaches to try to improve our inferences. And you just mentioned an example of that, right? And there are many more, right? And I think we should pay a lot of attention to that. I mean, some apply to some contexts, others have a more limited application, which is totally fine, it's expected, right? But we should pay more attention to that. So the answer is yes. In terms of post hoc analysis, hopefully we can agree that there are different schools of thought in the statistical world, right? So I want to be careful not to offend either camp, right? So I'm going to navigate this very carefully. But I think estimating the type S, type M error is a really good idea. Some of these quantitative approaches also require a large sample size. They're very data hungry, right? So we also need to keep that in mind, which connects to the idea of underpowered studies that I've been talking for the last hour. So yeah, the answer is yeah, we should embrace that. The other thing I wanted to say is that, and I'm biased with what I'm about to say, and it's mostly based on my experience, right, as a researcher, I think that we need to create opportunities to share these new methods. We tend to think that, oh, well, grad students should be trained on statistics and experimental design. Of course, right? But old people like me should also be trained on statistics and experimental design all the time, because their methods are coming live, right? That many of us are not even aware they exist. And that has a lot of implications for the whole research endeavor, because these more senior people, right, are the reviewers. That then, you know, if you decide to calculate the type M and type S errors, right, my reviewer might, what are you talking about? Yeah, just do a t-test, everything will be fine. You know, I'm exaggerating, of course. Right, so the learning has to be community-wide, not just that cohort of the new scientists that are coming up, right? Yeah, but that's a different sort of thing. Thank you, Kaya, for that great question.
I think I'm next. Please don't be shy. This is a topic that my impression over the years is that because people feel like they don't fully understand it, they don't engage. But if you think about it, for most fine seminars that you've attended, there's always a good chunk of the talk that you don't understand, right? You just cannot simply cover all the expertise presented in all the talks we've had. So some in the audience who are still here have suffered my hammering on issues around p-values. So I have to say that I'm so happy that Esteban has joined us and is presenting, as he opened up the talk, discussing the frequentist approach that I have so many times tried to move away from. So I'm loving it that once again in FINE, we can really be discussing different views, different approaches, and understanding that any of these, as you said, Esteban, this is just one, I think you call it framework, right? And all of these can give you something valuable if you understand how you use them. So I have a couple of comments that I would like you, in the spirit of continuing the conversation, just comment on. For the small audience we still have, and I think that is what worries me, that we're not getting enough people to realize this is important. That's why I share the paper that I shared by Smaldino and McElrath in the chatting window, the natural selection of bad science. Some of my young colleagues are saying, why bother? Just have a catchy title and either Nature, PNAS, or Science will pick it up. It makes a lot of money through tweets and blah, blah, blah. So that's worrying. Now, going to the first part of the talk, you presented Lacan's work, and I need to go back to him. But he's building a straw man, because the first two papers you mentioned, the one by Valentin Andrein and Greenland, those guys several times in the Nature article are saying there's nothing wrong with the p-value. So coming back and saying there's nothing wrong with the p-value, well, that's what they're saying. I mean, that's what we are all saying. It is not the p-value, it's the dichotomous decision, thoughtless, meaningless, because it is what it is. I mean, it is thoughtless. People are not thinking whether they should use 0.05 or 0.1. That's what I mean by thoughtless. And it is meaningless because people are not thinking about effect size. Therefore, I cannot judge what it means. So that's one. And then going to the end of your talk, I really liked that you're saying we have science inference in that first flow chart. And you finished talking about statistical inference. And I just want to be sure that we're all on the same page, that the last few comments of yours are about statistical inference. But as you well said, that's a tiny, tiny portion of what we should be doing as scientists, which is making scientific inference. So these are just some thought reactions to your presentation. I want to hear more from you since you've been thinking about this.
Yeah.
Esteban he was telling me yesterday, when he was an undergrad, he was already doing an awful lot of stats at the University of Córdoba. So he has training that I don't. So I want to hear from you some more.
No. No, I cannot claim that training here. But I think so. We have frameworks to do our job, right? Everywhere, right? So going away from statistics, we have a bunch of theoretical models in behavioral ecology, a bunch of them, right? Okay, so you go learn about these models. Each model has tons of assumptions, right? So think of the Brewster-Scrounger game, right? There's a great book by Karen and Gerald Doe about the different types of Brewster-Scrounger models and whatnot. And what I love about that book is that there's a box, after the description of every mathematical model with the assumptions. And you read the assumptions and you go, okay, that's going to be interesting, right? The point is that when we go and test the models, I'm not talking about statistics, right? Are we paying attention to the assumptions of
No,
models?
we're not.
And sometimes we don't, right? And then we forget about checking the assumptions, right, of the models. We test them and we go, oh, we did not find what the model predict. Well, but are you meeting the assumptions? Because the model makes predictions based on those assumptions. If you change the assumptions, the predictions are going to change. So my point here is that when we're using these statistical frameworks, it's the same story. They're frameworks that make assumptions, right? The framework I mentioned to you today needs to use the 4 elements: type 1, type 2, p-value, and effect size. Oh, I just pay attention to the p-value. I'm sorry. I don't know what to tell you, but you cannot use the framework. Or you cannot make conclusions based on that framework if you don't tell me what your type 1 error was, what your type 2 error was, and where your effect size of interest is, right? So I think there has been a lot of information has been lost in the training of, including my training, of the generations in terms of statistics, right? Oh, just pay attention to the p-value. That's all. That's all you need. No, that's what I've been told. Honestly, that's what I've been told when I was in grad school, right? Yeah. So that's one thing. The other thing is, we don't need to use inferential statistics, right? Inferential statistics is to test a particular prediction, right? Or hypothesis prediction combo. We can use estimation statistics. That's totally fine. I have no problem. There are some superb examples in the literature on estimation statistics. I didn't talk today about that. We can use the descriptive statistics, right? That's totally fine. But one of the issues that we have, and I think we were talking a little bit about this with Kaya this morning, is that again and again, we have reviewers that have this said that, oh, you need to be testing a hypothesis. No, I don't. I don't. I can be doing a descriptive study and presenting the result, the estimates of my effect sizes. I don't need to run statistical analysis. Oh, no, you have to. No, I don't have to, right? Obviously, these are decisions that you need to make before you collect the data. You cannot make this decision after you collect the data because the experimental design is going to be different, right? My point to you, Eduardo, is that there is a wide variety. You can do Bayesian statistics. There's a wide variety of frameworks that you are allowed to use, but we have a bias to as a scientific community towards certain frameworks. The Frequentist inferential statistics framework. That's number one. Number two, we're not using it correctly, right? So it's a combined problem, so to speak.
Thank you. Thanks. Let me scroll down. Laura, you're next.
Esteban, thank you for that talk. And so could you, I guess I don't have a great question because, but I have an idea I wanted to, if you could speak towards. We were talking about two things earlier, well, long-term studies. how can long-term studies, now we're talking observational data, not necessarily experimentation, but long-term studies in the field help us to overcome some of these problems, if at all? And then I'll hold off for the second question.
Meaning,
If you have a long-term study, I mean, you know, you're going to generate different kinds of data, right? Different sample sizes,
I
different.
see. I see.
Yeah, I mean, is it reproducible? You know, and I'm asking the question mostly for people who are thinking of designing long-term studies, some of the benefits of it.
Right. That's a great question. So one of the things, and I didn't get the chance to talk more about it, is that we also have an obsession with the confirmatory method, and it's something that yeah, I'm not understanding because we need descriptive studies, right, to come up with hypotheses that we will then test with the confirmatory method. But if you have informal conversations with editors, oh, a descriptive study, oof, no, that's lower quality, which is not true. It's not true. Science needs both. Now, one of the problems that in terms of these questionable research practices is you have a descriptive study, suddenly you get a finding that is pretty amazing, right? And then you try to sell post hoc this as a hypothesis-driven study in the paper. So that's what harking basically means, right? But I think we need to publish, I'm sure many people will get upset with what I'm about to say. I think we need to publish more descriptive studies that in their discussion come up with hypotheses from the data analysis to be tested in the future. Those are the seeds of the hypotheses that should move the field forward.
But is that, Esteban, I want to build on that. What we just said, that of course I fully agree. And connecting to Lauren's question, say, and this is something we are we're working on these days. So if I have 27 birth seasons, 350 births spread out, could I use some of these resampling approaches to say, I'm going to sample random? So now, in thinking about sample size, but something that didn't come up in conversation, but I know you think about it, is population of inference, right? Tell me the sample size, tell me the. If I say, okay, you know what, I'm going to sample randomly from my 27 years of data for description, and then I'm going to random sampling, I don't know whether with or without replacement, to see how it goes. Not because this is going to be, this is it. But is that I don't know if where Lauren was getting at this. Is that one way of tapping on? Now, you do that, and people are going to say, oh, yeah, but you have changes between years. Now, if I came with a study of 3 years, nobody raises an issue. If I came with a study of 30 years and I started doing this, now suddenly there are problems. So is that one of the things, I don't know, Lauren, whether you were thinking along these lines, but could that be done? Is that a good use of multi-decade sample sizes?
Yeah, I think it is. Now, one of the challenges that you might have, and I would fully support that. One of the challenges that you might have when you submit these for revision is dealing with the biases we just talked about. So one of the things that people came up with in recent years, and I'm sure you've heard about this, is pre-registering your study. And I think that people think that, oh, pre-registrations usually apply to the confirmatory method. No. You can also pre-register your study if you're doing some kind of descriptive study. No problem, right? And I think what that does is, well, first of all, it allows you to, before you engage in the process that you just mentioned, Eduardo, it allows a peer review of that process and getting some very interesting insights as to how can you navigate that process so that is statistically rigorous, okay? So that's an advantage. The second advantage, so you can get feedback before you engage in that process. But probably, which, you know, we shouldn't be thinking about these, but these days, right? It protects you. It intellectually protects you to have that descriptive study pre-registered and then following that, because then when you find the results you find, whatever those are, right, as long as you follow those steps that you committed to follow, that study should be published. So it's a way of minimizing the biases against these sort of studies, the pre-registration, I mean, right? So I think there are some tactics that you can use these days to engage in that process. Now, I think the challenge is which journal would be willing to engage in those sort of practices, right? There are not very many journals in ecology and evolution that accept pre-registrations. It's changing, so that's a good news, right? More and more, but there are not very many. That's one challenge. The second challenge is to find the appropriate reviewers, right? Because some members of the scientific community are against pre-registrations, and they have their reasons. I respect those and whatnot. So you need to find a group of reviewers that is open to review a pre-registration, because it requires a little bit of a different approach relative to the regular paper. Does that answer the question?
Yeah, thanks. Sari, you're next.
Hello. Thank you very much for your interesting talk. I have to admit, I don't have really a question formulated. I have just some ideas floating in my head, and most certainly I won't be able to really convey them correctly. And I have most of these ideas of the p-value and all that comes from my collaboration with Eduardo, and I'm by no means really deep into this literature, but every time kind of meeting up or listening or reading about these topics, it seems like it's always presented as if, like, the easy example is like, oh, you do an experiment and you're having a control and a treatment, and then that's how you do, like, you calculate your power and like your effect sizes and whatnot in the example presented, not today in this talk or in the papers that you can read about. But we are behavioral ecologists studying wild animals in the natural habitat, in which there are a multitude of factors that are not in our control and limited by time and money, logistics, and all that. We potentially do the best that we can. And then we are trying to do good science, but then you read, it's like, oh, the p-value should be interpreted such as such. And we are presented with an example of, if you do an experiment, you have a control and a treatment. But we do not do experiments. We do not have a control and a treatment. Our science is slightly different, and I think it would be very helpful if the examples of how to correctly interpret the p-value and how to improve our statistical analysis are presented more in the way we do our statistical analysis, instead of just going to the easy example of an experiment with its treatment and a control.
Great point. Thank you. Thank you very much. Okay, so first of all, it's a great idea. Thank you. I'll probably need to think for some time to come up with strategies to implement it. But I promise I will. But the second thing, which is more short term, that I would say is, and I know this sounds weird, but bear with me. You don't need to run statistical analysis to do what you just said.
Sure, but
right?
Then I would also want to add here, and this is another like kind of floating thought, that is this conversation and all of your colleagues that focusing on the misuse of the p-value, isn't that a reflection of how academia works and the malfunctioning of academia, and that you're just trying to fix kind of a tab that is running like a leaking tap? while actually the picture is much bigger than that. And I'm not saying that it's not valuable talking about the leaking tap, but not publishing or only doing the descriptive analysis. You just said that journals don't necessarily accept that, or the reviewers and such thing, but that is the system in which we are working.
I agree so much with you that it's crazy. Yes, absolutely, right. Now I'm a very pragmatic person, right? So now obviously it's going to be difficult for each of us to make these wider community changes, right? But I'm hopeful because of all of you, right? So there are a lot of early career researchers here. You are going to be the ones that are going to enact those changes. Old people like me, we're just trying to help out a little bit, but we don't have much to do that. So many things, and I'm sorry I'm repeating myself. So you have constraints, you just define those constraints, right? There are a lot of things that you can do with other frameworks. So you can use the framework of estimation statistics. I didn't have a minute to talk about that. And it's a framework that is being used more and more and more and more. And that's a framework that uses a lot of effect sizes, uses the confidence intervals. One of the things that we have to be careful is that because some people misuse the estimation framework in the sense that, oh, okay, I'm not going to run a statistical test. I'm going to do these estimations with an estimating the effect sizes. And now I want to make a dichotomous decision. Whoa, 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 whoa. No. If you want to make a dichotomous decision, you have to go back to the framework that I talked about today, right? So my point is, you can embrace one framework, and as long as you use that framework correctly, everything should be fine, right? And I have started seeing really good papers. I mean, published in Nature, Science, PNAS, Proceedings of Royal Society, using estimation statistics and not running a single p-value, right? But we're not, when we do that, we're not allowed to make any dichotomous decision. We cannot say, oh, this is significant. No. That's the wrong framework, right? But we can say, okay, the difference between these two things, a group with mostly males versus a group with mostly females in your area is of 15%. I was talking today with Facundo, and he was telling me of one of his experiments, and he found this difference of 15% between, I don't remember exactly what, right? Well, 15%, that's a lot biologically speaking, right? That's pretty interesting. And you can make arguments about that 15%, right? How many p-values have you estimated? 0. Right? I think we just need to declare in advance what we want to do. Right? Hey, I'm testing a hypothesis. I'm going to use inferential statistic. Okay, then the rules that I talked to you today, you should probably try to follow them. Right? I'm using estimation statistics. Well, you don't need p-values. Right? Okay. Yeah, it's a short-term solution.
Thank you. Sorry, I want to comment some more, but we have 3 people. I hope you'll stay till the end, because you know that I fully agree with you. I want to support my young colleagues who are struggling with the situation, but I'd rather not keep talking now and just go on to the next. And Marie, you're next.
Yeah, thank you for a great talk. I'm a postdoc at the Universidad Católica in Chile. Oh. I have a question about, like, I'm moving kind of more to like the Bayesian statistics. Okay. And so then you already have like the uncertainty in your models and everything. But so the power analysis is not really necessary for when you opt to do Bayesian statistics.
I would agree with that. There was some interesting discussion maybe a few months ago about power analysis in Bayesian statistics. And I think some people said, well, there are ways of running a power, simil to, it's not exactly power analysis, right? The way we define it today, but a simil to a power analysis for Bayesian statistics. And that led to a lot of controversy. There are some papers here and there. So it's an area that I would say is in development, right? Eduardo and I had long conversations about Bayesian statistics, and he knows why even though I appreciate Bayesian statistics, why I tend not to use Bayesian statistics. And the main reason is because, and I'm being honest with you, I freak out about the priors.
Me too.
Right?
But I mean, what is to be freaked out? You're already deciding to function with a null of 0, regardless of all the knowledge you have a priori. I mean, just go with a silly, uninformed prior of 0, like you do with the null hypothesis.
Yeah, but I think one of the most important contributions of Bayesian statistics is that it allows you to think about those priors and to consider them analytically, right? Which frequentist statistics doesn't allow you to do it, right? So I think I have nothing against that sort of priors, nothing. So please don't misinterpret me. But I think the real potential of Bayesian statistics is to have these very informed or well-informed priors, right? And run those analyses. That's when you go, whoa, that's an extra dimension that frequentist statistics doesn't give me, right? But in fields, and every field is different, right? But in fields in which there is not enough knowledge to have really good priors, I'm running the test. I'm like, no, I don't feel comfortable, right? And I don't do it.
But Esteban, I loved. I mean, I really gonna go back and check on this idea that we have frameworks for everything. I really loved it, how you made us think about the frameworks we use in behavioral ecology. But when you were telling us, listen, you could go and check the literature for effect sizes on your topic of interest.
Yep.
And now you produce an estimate of the effect size, which is your prior. And now instead of having a null that is a silly null of 0, you say, you know what? My null is going to be what the literature says I should expect. And now you have, within a frequentist framework, you could have something that it's more informed, just right? I mean, you were suggesting doing this.
100%. Now, the interesting thing of what you just said, which you're 100% correct, is that some people flip that argument around and they say, because in frequentist statistics, you can do what you just described, getting the distribution of effect sizes, there's no need to use Bayesian statistics. I'm the messenger, don't, right? I mean, it's these statistical wars between Bayesian and frequentist, and I don't want to get into that. What is really objective is that there are several papers that have been published in which, with the same data set, they try to make dichotomous decisions with Bayesian and frequentist, and the results were the same. For dichotomous decisions, right?
Right.
Yeah, for dichotomous decisions. So I use Bayesian, great. I use frequentist, great. I just make sure that you use either the proper way. That's all.
Thank you.
Suleima.
Yeah, I also have all kinds of ideas floating around, and I think part of what I'm going to ask is perhaps similar to what Sari asked. And I don't even know how to ask this question. And so I think what I'm going to do is mention 3 different types of situations and see what you think, Esteban. So the first situation with regards to harking, what do you think? And I'll ask all 3 and then you can answer as as you want to. But the first situation would be somebody who sets out to test a particular hypothesis, and then in the midst of testing their hypothesis or at the end, when they do their analysis, they realize that, gosh, we could have designed this differently to test this other hypothesis. So that's parking. But what if the authors are very explicit and very honest about saying this is what happened, and at the end we realized that there was this other interesting information, and so consequently we're adding this to our paper. Okay, so that's question number one. The second question would be, certainly there are schools of research that actually think that having a hypothesis, a priori hypothesis that you're testing, narrows your vision, and so you're not able to see what else may be happening, which would then emphasize gathering data over long periods of time. Long, can be long, or shorter long. But then going back to the data they have gathered and finding patterns and interesting observations that they think are meaningful. And I think that's maybe what Sari was getting at. And then what is the third situation? Now it's escaping me, but it'll come back to me in just a second. Well, maybe you should answer those two, and I'll try to think of what the third situation was, and ask you after the first two.
Yeah, great points, Zulema. So I don't.
I remember. Wait.
Good.
Yeah, I remember the third one. This has always bothered me immensely, but I'm not sure whether it's just a bias that I have or whether there's something really wrong with it. I suspect there's something really wrong with it. Are people who, and I know people like this, who essentially look at enormous databases. Let's say somebody's interested in demographic effects of stressful situations. And so they might look at what happens with, let's say, sex ratios or birth order following wars or following famines, those sorts of things. But there's no hypothesis to begin with. There isn't even any general, other than maybe demographic changes.
She froze?
Yeah, she's frozen.
Maybe she'll come back. Let's give her a few seconds.
Come up with patterns and so.
Okay, Zulema, we
Did I
lost you.
freeze?
Zulema, we lost you for a few seconds. Can you just go back with 30 seconds to start where we lost you?
Okay, I don't know when you lost me.
I mean,
Okay.
Go back about 10 seconds, 15 seconds.
Okay, so people may not have any more specific question than, demographic effects after times of stress or conflict or famine or whatever. And so they look at humongously huge, like really, really big databases, and they start seeing patterns and so on. But then the way that those often get reported, it's almost like it implies a causality. And they may not, explicitly say, war or conflict causes these changes. It's more implied, but many readers will come away thinking that, when you have war or when you have a famine, there's more females born, afterwards, or more males born or whatever. But that looking at enormous databases and then I would say cherry picking, the things that they see that seem to form a pattern and that may be a correlation. doesn't sit well with me. And I'm wondering those three situations, what your opinion is, Esteban.
Okay. I might need some reminder on the second one, but I remember very well the first two. Okay, so let me start with the third one. So
yeah, this is very implausible.
Schools of thought.
Oh, I'm sorry. Can you hear me?
She froze again.
Okay, great. Oh, she froze again. Okay, sorry.
Why don't we wait to see if she hears you?
Of course.
In the meantime, I'm going to paste a link to a YouTube talk that I strongly recommend related to Suleima was discussing, which this guy calls the causal salad.
Argue,
It's
and come up with it.
Suleima, we lost you again for another 10, 15 seconds. So, but
okay,
that's okay. We're a small group. Don't worry.
Yeah. So, okay, the second one was when you have whole schools of thought, for example, or a framework of doing research where people actually think that having a hypothesis and testing hypotheses hinders research or hinders your imagination because it prevents you from, you're so focused on your hypothesis, it prevents you from looking at, broader interesting phenomena. But so they gather data with no hypothesis, and then they come back and they examine that data, and they're able to come up with interesting finding, interesting observations, interesting ideas that are in fact very valuable. And I said earlier that I suspect that that's probably the type of situation that Sari was referring to. And so you may have answered that already. I'm not sure.
I think the three examples that you provide are really great because it shows a diversity of approaches. And I think we need to, there is room in the scientific endeavor based on the literature for these different approaches, right? So in the first, I don't know which case now, so are we allowed to use the exploratory method by mining a data set, a long-term data set, and from that data set, in the discussion of the paper, coming up with hypotheses and predictions for others to test in the future? Yes, we are. Yes, we are. We just need to declare it, right? That's all. But yes, we are. Now, the challenge, as we were talking before, is are reviewers trained to accept the exploratory method in the same way as the confirmatory method? Unfortunately not. Mm-hmm. That's the problem, right? I have promoted this idea in the Animal Behavior Society long ago, that we need to train our reviewers, right? We really need to train our reviewers, and not just our students, the reviewers too, right? Okay, so that's one point. The other point was related, the third point, the causation, the sort of assumed causation, although there is no causation because it's correlation, right? That's a problem, and many people are trying to tackle that in different ways. I think it kind of goes back to the sort of theme we have been discussing, which is we need to provide better statistical training, better methods training to the whole community. And we're not doing a good job. Usually in undergrad, you just need one biostats class to get through. I mean, you can take more, but usually it's one. In grad school, also one. Sometimes people, well, you should also take, but the complexity of analytical procedures that we have these days to manage the sort of data sets that we're handling is huge. And one course, and I teach one of those courses, I promise you, it's not enough. We need to be thinking about at least 3 courses or 4 courses to really train our grad students to be able to navigate these different scenarios, right? Exploratory method, confirmatory method, a causal inference. Eduardo and I were talking about these the other day. There are quantitative approaches to try to get an idea of, well, how much causation can I infer from the analysis I'm doing? You know, how should I plan the analysis in order to try to get closer to causation and so forth. There are very well defined procedures to get there. Are we following these procedures? No, we're not. We're not, unfortunately, right? But they are there. What was your first point? Just to make sure I addressed the second and third.
Yeah, I think you addressed the first, perhaps somebody who essentially does harking, but is honest about it.
Oh, yeah,
and starts out.
Yeah, that's a
by
good one.
saying so.
Yeah, we just need to be transparent about it, right? So we came up with this hypothesis. We were running the analysis, and oops, hey, it's a completely different thing. And actually, this might be a different thing. We need to report that as such. We came up with this, and we're testing this post hoc, which has a lot of statistical implications to test that post hoc, right? And actually, remember, this is a kind of sad story, not so sad, but one of my grad students years ago, we were testing hypotheses about the different visual fixation mechanisms in birds. So, you know, we had 2 or 3, whatever, right? And we developed the test and da. So my student ran all those tests, and the birds did not fit any of these fixation mechanisms that were reported in the literature. But then my student said, oh my gosh, this is a new fixation mechanism. And my student very correctly reported that in the paper as, hey, this is a post hoc thing. We didn't come up with these, so we're going to test these in the results section. The reviewers were screaming, how dare you do that? This should be in one of the original hypotheses. If you don't include this as one of the original hypotheses, we're not going to publish that. And my student, rightfully so, say, okay, I'll go to another journal. I'm not doing this. Yeah. Right? Yeah, and it took a while until one journal said, no, yep, we're going to publish this, you know, and we're going to report it like you want, right? Again and again, we need to train reviewers. Yeah.
Thanks.
Thank you. Let me see if we have, I know we have a question from Laura and then from Kaya. Please, we give priority to those who have not asked questions, so don't hesitate. But in the meantime, while you ponder if you want to ask something, Lauren, I believe you're next.
So this is a great discussion. One of the things that influenced me early was Kay Holekamp's paper, and I think it was 83, where she tested 10 hypotheses. And, you know, ever since I really ingested that paper, you know, I've really tried to do multiple hypothesis testing, which I think is critical because I don't do experimental work. And yet it creates problems statistically, especially now when you're thinking about linear mixed models that we tend to use, where you have lots of variables going into the model, and that gets to the sample size issues. Could you sort of speak to that, the challenges? And again, I'm not asking a question necessarily, but maybe getting it out there so people who are thinking about these things have a resource. Could you talk to the challenges about, you know, statistical models that are, you know, more and more we're needing to build these big models, and then we do AIC to figure out which one's best. But the big models are generated from hypothesis testing, multiple hypothesis testing in some ways, right? Could you speak to the challenge and the importance of that and how to navigate that issue, I guess?
So that's a great question. One aside, because of what you said, and a paper or papers that we read in the lab a few years ago, months ago, I don't remember, which is that the AIC in the context of inferential statistics, in order to select your model, right? One of the things that we learn is that we shouldn't use that unless we're trying to develop a predictive model, right, which is a completely different, you know, statistical procedure that you need to follow, right? So, and you know, we did that multiple times. So we have multiple models, AIC done. And what we learn is that statistically proper, right? We shouldn't do that. You know, the IC is reserved for these predictive models. But that's an aside. That's what we learned recently. All right. So right. One of the so if you have a general linear model, you know, think of an ANOVA or a linear regression or even a multiple regression, and you want to run a power analysis, no problem, right? You can use GPower, you can use a code in R. There are packages that do that for you. Yeah, it's pretty okay. When you have a linear mix model that you would like to use eventually, and you're trying to run a power analysis, you're not going to have a pleasant afternoon. You will not. Because you're going to hit a wall and it's going to hurt a lot. And it has been like this for some time. And one of the reasons is because using these sort of formula-based power analysis limits your ability to run any kind of power analysis for these complex models. The solution to that, I mean, I'm not saying it's the solution, but one of the alternatives to solve that is simulation-based power analysis. Because what you do then is you say, you know what? I can simulate data with the levels of complexity of my system. I can do that. And then what I'm going to do is that simulation-based power analysis. I'm going to run, you know, you need to use variation in your data and, you know, simulate multiple data sets. And you're going to run the statistical analysis 1,000 times. And then you estimate how, depending on, you know, you can vary your sample size, right? In this case, your sampling units might be individuals, marked individuals, right? So you can manipulate that. You can also manipulate by simulating those data, the number of times that you sample each individual, right? You can vary the number of times you sample individual, and then you get your power analysis done by basically running your mix, that very complex mix model, right? Multiple times. So that's the way we also use mixed models like crazy in my lab. That's the way we're going right now to learn more and more about this simulation-based power analysis, which simplifies tremendously that a priori power analysis. But the most important thing in my view is it reflects closely the data structure that you are going to be dealing with.
Interesting. Can I just follow up before?
Yeah, of course.
There's a follow-up comment. I think this relates to the p-harking or the harking conversation.
Mm-hmm.
And I think it ties back to what you were saying about the review process playing a role in all this. I mean, I get this, I'm dealing with this right now in a revision that's been a revision that's been a revision. It's going on and on and on. Too many hypotheses, models were overestimated, new hypotheses emerged post-review. And so the review process can actually help you to generate new hypotheses. It's a form of harking, right? But I think it's actually a beneficial form of harking when other people help you to think about what you can get from your data set, right?
Well, so it's not harking as long as you report what you just described.
Yeah.
It is harking when you, so you have 10 hypotheses and then you come up with 2 new ones after the data analysis. It's harking if you say in the introduction, we had 12, that's harking. But if you say we had 10, and then you're analyzing your data and then you get to the results like, hey, this could be a hypothesis, right? You cannot test it with your own data set. You have to be careful, right? But you can analyze it in that sense. That's not harking. So the harking component is how you report it.
Got it.
That's when it becomes harking, right? Which, you know, what some people say is, hey, I had 10 and then 2 came up post hoc, and then you write in your introduction, we had 12. It's kind of a lie because you didn't have 12, right? So that's what people that's what people consider a questionable research practice now.
Thank you.
So it's already 12:55. I'm going to take Kaya, who has one more question, and I think she's the last one. I'd love to continue discussing this, and I'm hoping that you'll take at least 2 minutes after Kaya finishes to see if there's interest in some of us seeing if we can create an additional time space for whatever it is that we want to do. But Kaya, you're last, not least. Go.
Okay. I was just thinking about the discussion between Eduardo and Esteban on using Bayesian statistics when we don't have very much information on priors in our field. And I'm just wondering with how that's compounded by the research bias in general, which exists in our field also of reporting significant results and underreporting non-significant results. If we are trying to use, not very much information in our field to set priors, and that information is potentially heavily biased, I wonder if it is better to do a null hypothesis or use the bad information. And maybe there's a way to estimate how bad it is, using curves of the p-values for whatever data set or prior knowledge you're using. But I guess I'm just putting that out there and seeing what you think about that.
Yeah, it's a really good point. I'm not an expert in Bayesian statistics, so I just want to make sure that you know that, right? And as I said before, I think the challenge is the priors and, are they informed enough and whatnot. I'm part of a thesis committee of a student here at Purdue, and the student wants to use Bayesian statistics to analyze a big data set. And I think the student came up with a very interesting idea, which is, it's a huge data set, right? Huge. So the idea is, I'm going to sample that data set without replacement. And what I'm going to do, and it is so huge that sample size wise is good, that with that, I'm going to develop priors for the system. And then I'm going to use those priors that I develop and test them with a different data set using Bayesian statistics. And I thought, I was like, wow, I'm not an expert, so I don't know if that's appropriate Bayesian statistically or not. But I thought that's a very interesting idea, right? Because you're developing your priors based on that system, right? So I'm not sure if I'm answering your question, but hopefully you understand that it's challenging for everybody, right? And I don't think that there is very explicit guidance as to what we should do. But having said that, I think if we are transparent in the way we explain that, I think if we share code, share logic, share data, share everything, I think that will improve the trust in the process, right?
Yeah.
That's my
Well,
take.
you don't need to ask for trust if you share, because trust implies that you have to trust me. I don't want you to trust me. This is the data. Do it for yourself. So actually, you're removing the need to trust, which is something we should be removing in science. It should not be about trusting. That's religion. And if you provide everything, if 10 years from now something comes up and said, "Oh my, I can't believe they were doing this a decade ago," okay, do it again.
But,
So I'd like to stop. Give me a second, Zuleyma. It's already 3 to the hour. So I don't know how many people we have in our YouTube community watching us live. Remember, the Zoom meetings are open to everyone. You don't need a passcode. You can join us in a minute. But if YouTube is your choice, you're welcome as well. I'm going to stop the live streaming right now. And to those of you in YouTube, we'll see you next week. Those of us in Zoom, we can take a few more minutes to continue a conversation. So see you next week. No, hold on. I almost left the Zoom meeting. I need to stop the struggling. Where are we?